Hello and welcome to this video lecture on Chapter 2 on genes and genetic diseases. The first topic we're going to discuss is DNA, and I hope this is review for you, but we have nucleotides that are DNA subunits that are made up of a deoxyribose molecule, which is going to be our sugar molecule. We have our phosphate, and then one nitrogenous base over here, which we can categorize within pyrimidines. That would be our cytosine and thymine, and then our purines are going to be our adenine and guanine. And we know that DNA is this double helix model. And so it appears twisted like a ladder, and then we have these chemical bonds that are taking place that are creating the rungs. And so we know that A is going to triple hydrogen bond with T, and G is going to double bond with C. And the reason DNA is so important in our cells are to direct synthesis of our proteins. We are going to see that they're made of one or more polypeptides, and we know the smallest subunit in a protein, or rather, let's say they're the building blocks of protein, would be our amino acids. And if you remember, these are the 20 amino acids that we can utilize to build a protein or a polypeptide and then pull it together as a larger protein. And it's directed by a sequence of bases. So let's go back to the beginning. We know that DNA is going to have to replicate itself, and then during a process of transcription, we are going to copy that DNA in order to produce a temporary strand, which we call mRNA, messenger RNA. At this point, we're going to leave the nucleus because all of this takes place inside the nucleus of a cell, and we leave that area to go into the cytoplasm here, where we will have a ribosome read that mRNA strand with the bases, the codons. Remember, those are in pairs of 3. And we are going to bring over tRNA molecules which have a amino acid bound to it, so that we can start reading and linking these amino acids together in order to build a polypeptide. And of course, there's a start codon to know when to start reading and building this polypeptide, and there's a stop codon to end it. So let's start off with that DNA replication that I mentioned in the nucleus. We need to untwist and unzip the DNA strand, and we have a single strand that acts as our template. And so we're going to start doing some complementary base pairing by an enzyme known as DNA polymerase. And we, of course, know our pairings. We mentioned that in the previous slide. And so sometimes when we're reading this, we can have mutations take place. These can be any inherited alteration of the genetic material. When we mention chromosome aberrations, these are changes in the number or the structure of our chromosomes, and they could be caused by errors in cell division or exposure to radiation or chemicals. So there are a couple different types of chromosome aberrations. We have aneuploidies, which means that we have a change in the number of chromosomes. Most of us know that there are 46 chromosomes within a cell, so you can have one extra or one fewer, and we're going to talk about this in a couple of slides. And then we can have structural aberrations, where we have changes in the structure of the chromosome, such as deletions or inversions, translocations or rings, and then isochromosomes, where the chromosomes arms are genetically identical. So we'll get into all of those examples in just a little bit. But when one of the things that can happen is a base pair substitution. So this is kind of an umbrella term, and it basically just means that one base pair is substituted for another. So sorry, my screen is a little bit small here. I'm trying to read these letters. So here we have our normal DNA. You could see that in this region we have our AT and then GC. And then as we are creating our mRNA strand, we of course want to pair the proper nucleotide here. So with our C, we want G. And so when we have a base pair substitution, that is called a missense mutation. That means that we are going to produce a single amino acid chain. So notice how here, when we code it properly, we are going to create the amino acid serine to go in the sequence. But over here we see that this has switched, and instead we put an A here, which then is going to change our amino acid, and that amino acid is asparagine. Whereas our nonsense mutation that takes place, you can see here is our normal set. We are going to code for tyrosine here for our amino acid, but when we switch that nucleotide up, it is going to code for a stop codon, which means it ends that polypeptide early rather than possibly continuing on to add another amino acid, which we see here. So we don't get anything over here because we stopped it. We also have something called a frame shift mutation, and this is when we have an insertion or deletion of one or more base pairs, and this can cause a change in the entire reading frame. So once again, here we have our normal process where we created our mRNA strand. You can see the amino acids down over here. When we have the frame shift, we see that instead of AT here, we have ATCG. And so that then is going to change the corresponding nucleotide here on our mRNA strand. We have added an extra set here. See how it kind of like squeezed in between the two ATs? So we had an insertion here, which then not only changed this amino acid here, but it also altered all of the codons downstream, and in turn changed all of the amino acids that come after that. And we could get some spontaneous mutations. This is a mutation that occurs in absence of exposure to known mutagens. And we have areas that we call mutational hotspots. These are areas where the chromosomes have high mutation rates. They undergo a lot of changes more frequently, which kind of increases the chances of us, let's say, making a mistake. And this can be more apt to happening if we have things like environmental exposure to toxins and radiation and all of that. So you may be wondering, what does that term mutagens mean? It means that we have agents that are known to increase our frequency of mutations. So examples would be things like radiation and chemicals such as nitrogen mustard, vinyl chloride, alkylating agents, formaldehyde, and sodium nitrite. So when we talk about radiation, we know it's a powerful, like, physical mutagen that's damaging the DNA. It does this in a couple different ways. You may know that we have two major types of radiation, such as ionizing radiation. That's going to include things like your X-rays and our gamma rays. And what they do is they break the DNA strands apart and cause base modifications that can then lead to chromosomal aberrations. So if we use a gamma ray, it can cause a double strand break within the DNA. We also have non-ionizing radiation. This can include UV light, and this could induce the pyrimidine dimers to basically cause mutations during replication. And we know that, of course, UV rays from sunlight could cause skin cancer. We already mentioned some of the chemical mutagens. I'll just mention some of the ways in which they can cause damage to DNA. For instance, the nitrogen mustard is an alkylating agent, and that can add alkyl groups to the DNA, which causes cross-linking and mispairing during replication. The vinyl chloride is a known carcinogen, and this can form DNA adducts and induces more point mutations. And this can lead to things like liver cancer and angiosarcoma. Our alkylating agents transfer the alkyl groups to DNA bases, and that could lead to incorrect base pairing and the strands breaking. So we see this a lot in chemotherapy. We use it to kind of damage the DNA of our tumor cells, but it can cause secondary malignancies. And then we have formaldehyde, and this causes the DNA protein cross-linking to lead to replication errors and makes our chromosomes unstable. And so this can be linked to things like leukemia and nasopharyngeal cancer. And lastly, we have sodium nitrite. This converts the nitrosamides in the body, which can deanimate the DNA bases, specifically A, G, and C, and this is typically associated with gastrointestinal cancers. All around not good things. So let's switch and move past DNA replication and talk about transcription. So our RNA is synthesized, like we said, from that DNA template, and we use RNA polymerase in order to bind to an area that we call the promoter site and start creating our mRNA strand, messenger RNA. And then it's going to detach, and the mRNA strand will move out of the nucleus and into the cytoplasm. So let's go ahead and take a look at that movement. So here is where we created the mRNA strand. Then we go through a process called RNA processing. Here we are going to see that we have these different parts, the exons and the introns, and we want to remove the introns. And once we go through that processing, we leave the nucleus and go into the cytoplasm, where we can move into our next process, which is translation. Now that we're in the cytoplasm, we are going to have protein synthesis take place by using our ribosome here and reading our mRNA strand. As the strand moves into the ribosome, we are going to bring in these tRNA molecules, which, as you can see, when they are coming in, they are bound to an amino acid here. We know which correct tRNA to bring in based on reading the codon. So the tRNA here has the anticodon. So let's say that this mRNA strand said AAU. That means this should say UUA. That would be the anticodon that matches this codon. And so that is going to have a specific amino acid attached to it. And as they come in and shift down to this middle spot here, we are going to bind the amino acids, creating peptide bonds and growing our polypeptide chain to eventually get our long polypeptide chain, which we can combine with other polypeptides and create a large protein. So I just want to go to this slide that shows that process taking place. It is moving through pretty quickly, but you could see how those amino acids are all attaching to one another until we reach the termination signal, where the ribosome is then going to dissociate from one another, the small unit from the large unit, and the polypeptide chain will be released into the cytoplasm. Now here is a question for you. Which of the following statements is true? A, RNA is double-stranded. B, DNA is replicated in the cytoplasm. C, RNA contains the same bases as DNA. Or D, a mutation is an inherited alteration of DNA. So let's think about A. If we have RNA, we know that it is actually single-stranded. So this one is a false statement. B here, DNA is replicated in the cytoplasm. Now, our DNA lives in the nucleus, so it doesn't make sense that it would be replicated in the cytoplasm. We would want to do that in the nucleus. So false statement. C, RNA contains the same bases as DNA. Now, we didn't directly talk about this in this lecture, but hopefully from a previous course, you know that there is a set of bases that does not cannot be found in either of these, or not either of these, but like they could only be found in their own type of nucleic acid. So for DNA, that's going to be our thymine, whereas RNA is going to have uracil. So we will never find uracil in DNA. We'll never find thymine in RNA. And so that's a false statement. So then we end up with D. A mutation is an inherent alteration of DNA. That is a correct statement. Now let's talk a bit about chromosomes. So we have somatic cells that contain 46 chromosomes. You can see that pictured over here. Of course, there's not 46 of these, but you get the point here. They're kind of paired together. So they're 23 pairs of chromosomes. And we call these diploid cells, and they're formed through mitosis. Now we have these other cells, our gamete cells. Think of them as the eggs and sperm. They're going to contain 23 chromosomes, and these are known as haploid cells. And we find one member of each chromosome pair within these gamete cells, and these are formed through meiosis. So you could see that process of meiosis taking place here in a simplified version, and here we see mitosis, where the somatic cell is basically going through cellular division here to make 2 copies of itself. So our chromosomes, we have 23 pairs like we said, and so pairs 1 through 22 are called autosomes. They are going to be found in both males and females, and they're 2 members that are virtually identical and thus said to be homologous. The last pair, the 23rd pair, is going to be known as our sex chromosomes. And so in females, we will find XX pairs, so they're homologous, whereas in males we find XY like we do in this picture, and these are non-homologous pairs. And this image that we're seeing here is known as a karyotype or karyogram, and it's just an ordered display of these chromosomes. You can also see in this image what the pairs kind of look like when they are put together, and there's a centromere in the center over here. All right, so our chromosome aberrations. When we have our chromosomes that are healthy and normal, we have the right number, they are known as euploid cells. Now, our haploid and diploid cells are euploid forms, even though we know that haploid is 23 chromosomes and diploid is 46 chromosomes. But that's normal depending on if we have our gamete cell or our somatic cell, right? So when we have a euploid cell that has more than the diploid number, it is known as a polyploid cell. So this is not something we want to see. Here we have an example of a triploidy, meaning a zygote. Remember, a zygote is a fertilized egg, so we should have 46 chromosomes there, but they have 3 copies of each chromosome, and so we have a total of 69 chromosomes. This is known as a tetraploidy, where we have 4 copies of each and end up with 92 total chromosomes. Of course, neither of these fetuses will survive because we do need that 46 number of chromosomes. Now we can have a couple more, a couple less to survive, but this will not work. All right, so we have this term aneuploidy, and this is a somatic cell that does not contain a multiple of 23 chromosomes, and we call this non-disjunction. You could see that labeled over here as well as down over here, where we produce some gametes that have 2 copies of a given chromosome and others that have no copies. So you could see the empty cell over here. And when these gametes unite with a normal haploid gamete, the resulting zygote can either be monosomic or trisomic. And what that means is when we have a monosomic cell, it means we just have 1 copy of any chromosome. So you see the single chromosome here in this cell, whereas with trisomic, we're going to have our 3 copies of 1 chromosome. Now, having the monosomy cell here is lethal, but infants can survive if we have the trisomy of the chromosomes here in the cell. So it's better to have extra chromosomes than less chromosomes. And here's a little description on non-disjunction, even though we talked about it in the previous slide, but it's that failure of having homologous chromosomes or sister chromosomes separate normally, like we see over here during meiosis or mitosis. So here we have the trisomy and the monosomy, and here we have that taking place during meiosis 2, where we end up with a trisomy here and a monosomy here. Now let's talk about the autosomal aneuploidy, in which we have a partial trisomy. So this is only an extra portion of a chromosome that's present in each cell, where the consequences of having this partial trisomy isn't as severe as those that are complete. So it may only occur in some cells of the body. And so in the cells where the chromosomes are affected, we call that chromosomal mosaics. In this image over here, we can see a 13-year-old patient showing to have a long face with a down slanting palpebral fissures. She also has strabismus and high nasal bridge and a prominent jaw. We can also see on the ear a prominent antihelix and a dysplastic concha. Here we see the right foot that has an overriding third toe secondary to the short third metatarsal bone, which we can't see from this image, but through an x-ray. And then we also see a bilateral vertical plantar furrow on the feet here, and it's hard to tell in the photo, but there is a short overriding third toe that's hidden in this view. Another example in which we can see this is in Down syndrome, pictured in this with this patient over here. This is a mosaic trisomy 21, where some of the cells have 3 copies of chromosome number 21. So as I mentioned before, this is the best known example as an aneuploidy, and it's in about 1 of 800 live births. Some of the effects are going to be intellectual disability, low nasal bridge, epicanthal folds, protruding tongue, which you can see pictured or illustrated here rather, and low set ears and poor muscle tone. We'll also see possibly a gap between that first and second toe, and broad hands with short fingers, and possibly abnormal and excessive facial fat distribution. And we know that in women who are above the age of 35, we have increased risk for this trisomy 21. Now for our sex chromosome aneuploidy. One of the most common is trisomy X. This is a female who has 3 X chromosomes, and so they're going to have a total of 47 chromosomes instead of 46. So symptoms here are going to be variable. It can lead to sterility, menstrual irregulation, and or intellectual disability. We can see some facial features may change, such as having those epicanthal folds that we saw in Down syndrome, and something known as hypertelorism. And so that is going to be a condition where the distance between the 2 eyes here are increased. You can see here is a normal distance, whereas as we continue on down, you can see that hypertelorism. And this is a couple different patients that have this triple X aneuploidy, and you could see some of the features within their faces. So here we see a 2-year-old girl, here is a 9-year-old girl, and I'm not quite sure the age on this, 19 years old on this patient, who all have trisomy X. Next, we're going to discuss Turner syndrome, and we have a single X here because this is a female that has just 1 X chromosome. So their characteristics or clinical features are going to be underdeveloped ovaries, meaning that it can lead to sterility, having a shorter stature, around 4'7", webbing of the neck, which you can see illustrated over here. Also edema, underdeveloped breasts and wide set nipples, and a high number of aborted fetuses. And these are some features in another patient here, where over here what we see depicted is having the hypertelorism, so a little bit more distance in here, and the epicanthic fold and down slanted palpebral fissure. In this picture, we can see a shield-shaped chest with widely spaced nipples and poor development of the breast. Over here, we see a severe neck webbing and a little bit of a lower posterior hairline. And over here in the hands, we see abnormal left hand creases and an absent proximal flexion crease in the fifth right finger. So if you can see this, I know it's a little small. There's no crease in here. Next up, we have Klinefelter syndrome, and this is known as XXY because this individual is going to have 2 X chromosomes and 1 Y chromosome. So the characteristics here is to have more of a male appearance, also a development of female-like breasts, having smaller testicles, and that typically is going to lead to sterility, and having sparse body hair and longer limbs. You can kind of see how this is a little bit longer, moving past the hips and down about midway of the femur. So some individuals too can have, like we said, the 2 X and 1 Y chromosomes, whereas others can have 3 X's and 1 Y chromosome. And abnormalities are going to increase with each X that gets added on here. Now let's talk about some abnormalities in the chromosome structure. So we know that obviously chromosome breakage is not great. That is when we're going to have a physiologic mechanism that does not function in repairing a break. And the break can heal in a way that alters the structure of the chromosome. So if we're exposed to things like clastogens, that can increase our risk of these types of breaks, and that could include things like ionizing radiation, chemicals, and viruses. If we have a deletion, that could be due to a breakage or loss of DNA. So you can see over here in this figure that we have A through H, which is fine. And then here we have the continuation of the alphabet. And then we have a break occur, and we see that we have lost this DNA content in here. So it's been deleted. The most common example of this is a disease known as Cri-du-chat syndrome. This is a French pronunciation, and it literally means cry of the cat, and it describes the characteristic cry of an affected child. So they have a deletion of the short arm of chromosome 5. So I have something pictured here to depict that. And the result is having low birth weight, severe intellectual disability, and microcephaly, which is a smaller head size. You could see that there are other features of this, such as having a small chin and having low set or abnormal ears and so forth. And this is just an image of an actual patient who is affected with this at age 6 and age 16. And sometimes we can have duplication, meaning that we have the presence of a repeated gene or gene sequence. This is less serious because, as we mentioned before, it's better to have more genetic material than less, like we saw with the deletion. And duplication in the same region as Cri-du-chat causes intellectual disability, but less serious physical abnormalities. So this figure over here depicts how we have normal crossing over take place. So you can see over here that we have a shifting from like the capital letters to the lowercase letters. Normal crossover. I kind of wish they color coded this a little bit better, but and over here we're seeing an unequal crossing over. Notice how these two legs are longer than these over here. And then we have inversions where we have 2 breaks on a chromosome, and it can cause a reversal of the gene order. So here we have A through G, and what happens is this segment C through E is basically inverse. So notice how it's kind of going backwards here, C, D, E, instead. And this usually occurs from some type of breakage that gets reversed during reattachment. This can also be called a position effect, where we have a change in a gene's expression caused by its position, and this could sometimes cause physical defects in people. Next up, we have translocations, and this is where we have an interchanging of material between non-homologous chromosomes. So as we saw with inversions, the carrier of a reciprocal translocation is typically normal, but if their offspring can have duplications and deletions, that may cause some side effects. And here we see an example of that reciprocal translocation. We see A through G here and H through O over here, where we are taking this section over and shifting it over here and moving C through G and placing it over on this side. And this is our result over here where we had that exchange. We can also see that pictured over here with the use of colors. And then we have something known as the Robertsonian translocation. And this occurs when fusion at the centromere forms a single chromosome. So I have an example of that placed over here, and it's typically confined to chromosomes 13, 14, 15, 21, and 22 because they tend to have short arms, and these chromosomes are really small, so they don't really contain a lot of genetic material. Their offspring, though, can have some serious monosomies and trisomies take place, and they're really responsible for about 3 to 5% of Down syndrome cases. Now, we do have some areas that we call fragile sites. These are areas on the chromosomes that develop distinctive breaks or gaps when cells are cultured, and an example of this is Fragile X syndrome, where the fragile site is on the long arm of the X chromosome. And if someone has this condition, then they are associated with having intellectual disability and second in occurrence to Down syndrome. And we do see a higher incident in males because they only have one X chromosome, whereas females have 2 X chromosomes. Now let's talk a little bit about genetics. So these are just some key terms to understand as we continue discussing about DNA and genetics and how that relates to mutations and disease. So first up is locus. You could see that pictured over here, and I like to think locus for location. It's the position of a gene along the chromosome. So here we could see that this locus is the position that the hair color gene is on. It is going to give us our phenotype of having black hair. And an allele is a different form of that particular gene at a given locus. So we can, for instance, have the gene here for blue eyes, but it could also be for green eyes or for brown eyes and so forth. And sometimes we can have a locus that has 2 or more alleles that occur with appreciable frequency. Essentially, this allows us to have more variation or alternative forms of a version of a gene so that we can have more variation, really. Now let's talk about the terms homozygous and heterozygous. Homozygous means that the loci on a pair of chromosomes have identical genes. So for instance, I'm just going to make this up, but let's say these B's represent brown hair and the lowercase b's represent blonde hair. So if we have 2 big B's here, that means we're homozygous for definitely having brown hair, whereas over here we are homozygous for having blonde hair. Whereas if we're heterozygous, that means that we have a pair of each type. So the big B, brown hair, and little b, blonde hair. So maybe we're going to have a variation here of light brown or something like that. And same thing over here. We have both types in here. And then we have the differences between genotype and phenotype. So genotype is really talking about the composition of those genes at the locus. We talked about homozygous and heterozygous pairs Notice how we have dominant and recessive. When we say something is dominant, that means typically when it's present, it is going to cause that phenotype to appear. So we use the example here of the flowers and their colors. So the big B is representing the dominant gene that brings out the purple color. So when we have heterozygous together, that big B is bringing out that purple color. It kind of overrides the recessive little b. But when we have two little b's that are homozygous, that means it's recessive and we are going to get that white color. We also have an example of an infant that is born with PKU genotype and is left untreated. They can develop intellectual disability, but if they are treated, then that child has an outwardly normal PKU phenotype, meaning that they will not have problems trying to break down the amino acid phenylalanine. And here's a further explanation of the terms dominant and recessive. This example is utilizing eye color. So we see the big B's are representing having a brown eye color, whereas the recessive two little b's are representing having the blue eye color phenotype. So in genetics, the dominant allele is going to be represented by a capital letter always, and the recessive will always be represented by a lowercase letter, and alleles can be co-dominant. All right, transmitting genetic disease. So these are the different modes of inheritance in single gene diseases. We have autosomal dominant, autosomal recessive, X-linked dominant, and X-linked recessive. So I'm just going to give a quick summary of each of these, but we are going to go into detail in the coming slides. When we say autosomal dominant, this can cause abnormal traits in offspring, even if only one parent has the gene, which is kind of what you're seeing over here. When we have autosomal recessive inheritance, that will not produce abnormal traits unless both of the parents have the gene. When we have an X-linked dominant inheritance, that means we have a person with an abnormal trait that has one affected parent. If the father, so in this instance over here, if the father has an X-linked dominant disorder, all the daughters and no sons will be affected. If the mother has an X-linked dominant disorder, then there's a 50% chance that each of her children will be affected, which is what we're seeing here with having 4 children, only 2 of them are affected. And then we have our X-linked recessive inheritance. Affected people here are usually going to be males with unaffected parents. So this is showing us a carrier. So even though she has the gene, she's not displaying the actual hemophilia. And if we are to have a male who's affected, then all the daughters will be carriers. The sons of affected males will be unaffected, and unaffected sons, which we see pictured over here, can't transmit the disorder. Unaffected male children of a female carrier, which we see pictured here, don't transmit the disorder. So we can utilize pedigrees to study specific genetic disorders within families, and it begins with a proband, usually when the first person is diagnosed. So like I said, we're going to dive into a bit more detail, starting with autosomal dominant inheritance. Our affected offspring are usually going to be produced by the union of a normal parent with an affected heterozygous parent. So let's think about what that means. Here we have a heterozygous affected parent. So remember, when we have that big capital letter there, we are going to have it carry the disease dominantly, so that's why this parent is affected, whereas this one has the recessive, and so they are unaffected. And what we see is that 50% of the children here are going to be affected, whereas the other half will be unaffected because they are homozygous. Now in the example below here, where we have 2 affected parents that are both heterozygous for the disease, we see that the homozygous is affected. This is rare, whereas we have the heterozygous here that are also affected. So 75% of their children will be affected, whereas here the 25% is unaffected with our recessive alleles here. Okay, and then we have recurrent risk. This is the probability that an individual will develop a genetic disease. For autosomal dominant, when we have one parent that's affected by an autosomal dominant disease and the other parent is going to be unaffected or normal, then the occurrence and the recurrence risks for each child are going to be 1/2, or think 50%. We have to remember that each birth is an independent event. So every child born, regardless of their siblings' outcome, has that recurrence risk of 1/2. This term penetrance refers to the percentage of individuals with a specific genotype who also exhibits the expected phenotype. And if we have incomplete penetrance, then that means our individuals with disease-causing genotypes may not show the disease phenotype. The genotype and the associated disease may still be transmitted over to the next generation. So an example of this would be retinoblastoma, which is an eye tumor that we find in children that usually is malignant, and it can demonstrate incomplete penetrance of 90%. So 10% are obligate carriers and don't develop the disease. And then we have this term expressivity. This is the extent of the variation in the phenotype associated with a particular genotype. And an example of this is neurofibromatosis type 1. Which is going to be a tumor. It's the tumor that is a soft dome shape, which you could see pictured over here, and is typically going to be benign. And factors that could cause this variability of expressivity are through modifier genes, meaning that the genes at other loci are influencing its expression. Also, environmental factors. So these would be non-genetic influences on gene expression, and then different mutations at a locus. So for example, the factor VIII gene mutation in hemophilia has a single amino acid change, then what we see is more of a mild hemophilia. So we can maybe think of this as like just a little bit of the TREPS here. Whereas if we were to have a stop codon mutation there, then we can have severe hemophilia. And you may have heard of this term epigenetics. What that means is that we have chemical modifications that are taking place to DNA that alter its gene expression without changing the DNA sequence. And the way this is done is through DNA methylation, so adding a methyl group here. The methyl group attaches typically to cysteine bases in what we call these CPG islands. And the CPG islands that are methylated reduces gene transcription. So you could see in this illustration that gene expression is turned off, whereas here when they're not methylized, then they are going to continue reading that. The impact here is that the phenotypic differences arise despite having identical DNA sequences. And this, of course, could be influenced through lifestyle factors, such as if somebody is smoking or not. And another way where we can stop the gene expression is through genomic imprinting, where one parent imprints or inactivates the gene during transmission of the offspring. Now, before we go on to talk about autosomal recessive inheritance, I thought we would talk through these Punnett squares really quick. So when we say affected parent, means the parent is carrying or showing the disease, and this is a heterozygous parent, so meaning we've got one capital letter, one lowercase, whereas here's our unaffected parent, and they are recessive for the disease. So what we do is we bring those down, so the capital A and little a come together. Because we have that dominant A, that means that we are affected. And same thing down here, we get another affected because we brought this A down here and the little a over here. Now when we do it over here, we see two lowercase letters, so they're recessive and unaffected. And lastly, if we were to draw this down and draw this across, we would also get a double lowercase, meaning they are recessive and unaffected. So this is, again, an autosomal dominant inheritance, whereas here, let's see what it looks like when we have autosomal recessive. So here we have our affected parent, again, heterozygous, and same thing over here. And so when we have the capital A's here now, they are unaffected because remember, this is autosomal recessive inheritance of the disease. So they're unaffected. If we were to go to, I think we're going to do this next spot over here, it'd be a capital A and lowercase a. So that is a unaffected carrier because we need both lowercase a's in order for someone to be affected. So that capital A is kind of saving them from being affected with the disease. Next one is going to be this lowercase a and the capital A. So we find another unaffected carrier. And then lastly, we bring these two down and we see the double recessive and they are affected. So let's talk about this autosomal recessive inheritance. In most cases, both of the parents are affected individuals or are heterozygous carriers. So if we look to our Punnett square over here, we can see in this case they're heterozygous carriers. So we can see that this person is unaffected. These people are heterozygous carriers like we saw in that previous slide, and then we would have 25% of the children or 25% chance of having a homozygous affected child. So this typically happens in people who are involved in consanguineous mating, known as incestual mating, or when people who are related by blood have a sexual relationship or marry. And the recurrence risk of the offspring is 25%, as we described in this Punnett square, and we see that males and females are equally affected. And an example of this are people that have cystic fibrosis, in which the cells are going to be affected and produce a mucus, sweat, and digestive juices, and this can cause these fluids to become thick and sticky and plug up tubes, ducts, and passageways, and typically will cause damage to the lungs and the digestive system. Now let's move into X-linked inheritance. We're talking about sex-linked inheritance here, and these are genetic conditions that are caused by mutations on the sex chromosomes. Most of the sex-linked traits are located on the X chromosome, and so they're said to be X-linked. So we have X-linked recessive, that's the most common, and X-linked disorders are typically expressed by males because we know that the females are going to have another X chromosome that helps counteract the abnormal genes on the other X chromosome. Something that's interesting is X inactivation. So when we have someone, let's start with our females. When we have an X chromosome in females, it is going to, one of them is going to become permanently inactivated early on in embryonic development. So it's about 7 to 14 days after fertilization. This starts to happen through embryonic development. So in every stem cell, so let's say this is taking place over here, in every stem cell, we are going to choose one X to become active, and the other X is going to become inactive. And in fact, that X chromosome that's going to be inactive is going to fall apart. And when it breaks apart, it is called a Barr body. So that's what's pictured over here. And so you could see with each of these, it's deciding, is it going to keep the X from the, let's say from the mother, or is it going to keep the X from the father? And from there on, these stem cells are going to go through mitosis and copy themselves. And so what we end up with is a mosaic somatic arrangement of cells within the female body. And so we can say that this inactivation is to be at random, but it's not fixed. And now let's talk about how we can determine one's sex. This is going to all be done through the SRY gene on the Y chromosome. So early on in embryonic development, when the gonads are developing around the 6th week of gestation, we're going to see that one copy of the Y chromosome is going to be just enough to initiate the process of gonadal differentiation for a male fetus. So basically, we are all females by default. But if you have a Y chromosome that is going to contain that SRY gene, then that is going to express for us the male gonads development. And SRY, by the way, stands for sex determining region of the Y chromosome. And it's located on the short arm of the Y chromosome. And so we can see this normally developing over here. Now, sometimes we can have a crossover take place on the centromeric side of the SRY gene, and this could cause it to move over and be found on the X chromosome instead of on the Y chromosome. And so the offspring that gets this gene could, on the X chromosome, could become a male XX person and may resemble a male that has Klinefelter syndrome that we talked about earlier. And offspring that receives the Y chromosome that is supposed, I don't want to say supposed to be, but that's female, is going to have a XY arrangement for their sex chromosomes. And these females will typically have gonadal streaks rather than ovaries, and so as a result they'll have poorly developed secondary sex characteristics. All right, our characteristics of pedigrees, we'll see that most of these X-linked disorders are going to be recessive, and the affected males can't transmit the genes to their sons, but they can to all their daughters. And so the females are going to become carriers for the disease, and the sons of the female carriers have a 50% risk of being affected. We could see some of the Punnett squares here. Let's start with that. At the very top here, we see a Punnett square for a normal male over here and a female carrier. So this is a X-linked recessive gene. And over here, we can see the results of a normal female. We've got our dominant alleles here with a affected father. And then lastly over here we could see the Punnett square for a female who is carrying the X-linked recessive gene. So she's a carrier, and the father here is affected by the disease since his X chromosome has the recessive disease here. And so a common example of a X-linked recessive disorder is Duchenne muscular dystrophy, and this is characterized by progressive muscle degeneration. Unfortunately, these individuals are unable to walk by age 10 or 12 years old. They also have effect to their heart and respiratory muscles, and death is usually by respiratory or cardiac failure, usually before the age of 20. And so we were able to identify the disease-causing gene on the short arm of the X chromosome. It's actually the largest gene ever found on humans that spans more than 2 million DNA bases, and it is there to code for the muscle protein of dystrophin. And that plays an essential role in maintaining the structural integrity of our muscle cells. So when that muscle protein, dystrophin, is absent, the muscle cell cannot survive, and so the muscle starts to deteriorate. Sometimes we have sex-limited traits, and this only occurs in one sex because of the anatomical differences. So an example would be an inherited ovarian defect. For instance, if a uterus is bicornuate, that means that we have these two tunnels instead of having that one big open space, whereas males that have testes, they are going to have defects such as cryptorchidism, where we have a failure of the testes to descend down into the scrotum. So here we see that the sex-limited traits are going to have more to do with those anatomical structures, whereas when we talk about sex-influenced traits, these have more to do with hormonal levels. So this can be a trait that can happen to either one of the sexes, but is more prone to happening in one sex versus the other. So an example of this would be male pattern baldness, where the gene for baldness is present in both males and females, but the expression depends on testosterone levels. So in males, the trait is dominant, so a single copy of the gene is going to lead to baldness, whereas in females, the trait is recessive. So we need 2 copies of the gene in order to have that trait expressed. Another example is autosomal dominant breast cancer, which is much more commonly expressed in females than it is in males. All right, another question for you here. Which term best describes an allele with an observable effect? Carrier, dominant, recessive, or homozygous? If you answered dominant, you are correct. They have observable effects. A carrier is going to be someone who has the disease gene, but we don't see it in its phenotype. Whereas recessive means that the alleles may be hidden and not observable. And then homozygous is when we have 2 alleles at a locus that are identical, whereas heterozygous, if you remember, is when we have 2 alleles at a locus, but they are different. Now let's talk about gene mapping. We talked a little bit about crossovers. This takes place during meiosis. You could see these chromosomes are coming together and exchanging their alleles. So that is known as recombination. And the frequency of recombinations is used to map distance between loci on the same chromosome. Here at the letter A, we can see allele pairs would be transmitted together when no crossover is taking place. So we have A1 and B1, and then A1 and B1 again, and then same thing here with the 2s, so that they kind of look identical here. But here at the letter B, we can see what happens when all 4 possible pairs of alleles can be transmitted to the offspring. So we've got A1, B1, A2, B1, so A1, B2, A2, and B2. So essentially we are able to form new arrangements, giving us more variety. And again, this is called recombination. And one of the goals that we set out to do is the Human Genome Project, where we wanted to create a gene map of all human genes. And luckily we were successful in that, and we're able to identify genes for 5,000 conditions. And this really helps with diagnosing conditions as well. We can also have phenotypes show up if we have polygenic traits, meaning that there's a variation in the traits caused by the effects of several loci. Essentially, we're saying that some traits are a result from several genes acting together. And you could see a multifactorial trait come about as well. This means we have variations because of both genetic and environmental factors. So we see that displayed over here where we have a couple different genes working together, and this can cause the grain color to vary in a gradual way from white to red. We can also see this multifactorial trait in things like height and IQs, depending on the nutrition that we're exposed to or the enriching learning environments that we are put within. Now, what we're seeing over here is something called threshold of liability, and this has to be crossed sometimes before a disease is developed. So this graph here happens to be one for pyloric stenosis, and that is a disorder that's characterized by a narrowing or an obstruction of that pyloric sphincter or the pylorus region, and doesn't allow contents to move into the small intestines. So therefore it kind of moves back and could cause things like chronic vomiting, constipation, weight loss, electrolyte imbalance, but we can easily correct this with surgery. So what does this threshold liability tell us? It means that an individual can appear normal and not have this problem until maybe we have some environmental factors that start to play in that push us closer to the threshold, and at that point we are going to see the individual become affected by the disease. Now, in the case of pyloric stenosis, females do have a much higher threshold before that happens when compared to males. And what we see is the recurrent risks of multifactorial diseases can change a lot between the different populations, because we're not really sure what environmental factors each individual is surrounded by. So the number of genes contributing to the disease could not be known. The precise alleles that are from the biological parents might not be known, and then of course the environmental factors will be different from one population to another. So that recurrent risk is going to become higher if more than one family member is affected, because there's probably a biological genetic component that is increasing that risk. And then as far as empirical risks, they're going to be derived via direct observation. And in order to do this, we need a large sample of biologic families in which one child has developed the disease and is examined. And then the siblings of each child are then surveyed and calculated into a percentage who also developed the disease.